well.
So I'll go ahead and get started. I'm excited to introduce Iliana Ruiz-Cooley. So she's one of our newest research faculty here, and I'm sure as you hear her talk, you will realize that she's a potentially great resource and collaborator for a lot of us here. She has pretty broad research interests. She uses stable isotopes to address questions in trophic ecology and migration of odontocetes, fish, and cephalopods. And she also uses isotopes to look at the effects of climate variability on ocean biochemistry and trophic food webs. She got her master's at, I translated it into English because I didn't know how to say it, the National Polytech University in La Paz, where she studied the foraging ecology of sperm whales and deep sea squid. And then she got her PhD at New Mexico State University. I think that was also studying foraging
Squid.
Squid, that was just squid. Cephalopod ecology. And then she moved to Southwest Fisheries, where she was working on jumbo squid, looking at their range expansion. And then she started a project using common dolphins as indicators of coastal ecosystem change. And then she's been working on something I think fairly similar with sperm whales, and I'm sure she'll be talking about that today. So.
Thank you, Gideon. Well, can you hear me? Okay, well, thank you for the invitation, and I'm really excited to show or share some of the results that my collaborators and I have been conducting in the California Current. I've been working in different ecosystems along the Eastern Pacific Ocean, but I thought it would be important to show you the work and the findings that we have we have found here in the California Current. So I'll be talking about 3 different species, the jumbo squid, sperm whales, and sharp-beak common dolphin. And at the same time, I would like to show you the application of stable isotopes, the advantages as well as the limitations. And let me see. Okay, I have the pointer in the right position. There we go. So we know that our planet is warming, and we need to understand how warming is going to affect ecosystems and communities. But also, we want to understand how communities respond to natural climate variations. And this is something important that many scientists have been trying to understand for many years, for centuries, actually for a century. So I'll be talking, as I said, about the California Current. You know about this system, so I'm going to talk only briefly about this. It's a highly productive ecosystem that can be divided in 3 regions, the northern, the central, and the southern part. It goes from 50 degrees north to the south part of Baja Peninsula, and we have to the east the North Pacific Gyre. And we have many scientists recovering information from this ecosystem for many years. Since the 1949, for example, we have the Calcophie program that monitors this ecosystem in the southern part of the Southern California. And also we have NOAA that conducts surveys to understand the abundance and distribution of cetaceans, fisheries, and also universities, and one of them, of course, Moss Landing Marine Lab, trying to understand aspects of the marine biology of these local ecosystems. So we know, and we have information that the ecosystems of the California Current undergo huge fluctuations in temperature and other variables. As you can see here, there's huge fluctuations in sea surface temperature. This is from the Calcophie record, as well as the PDO, the Pacific Decadal Oscillation. We have also information that the concentrations of oxygen has changed. Since the 1950s, there's a slight increase, and after the 1980s, there's a decrease in the concentration of oxygens in the water column. So what does that mean for the species? How the population of species or the communities respond to these changes in the physical and chemical environment? And we have a very good amount of information regarding the changes in the abundance of distribution of species. We know that there's huge fluctuations in relation with the PDO, for example, anchovy and sardine. And also we know that there's strong changes in relation with El Nino. And one of them, as you know, and you're familiar with this, is occurred in the 1997-1998 El Nino that is considered the strongest Nino of the past century. So this species is endemic of the Eastern Pacific Ocean. It grows very fast. It goes from larvae to 1.5 meters. They can live about 1 year, and there's some information that tells us that they can live 1.5 years or maximum 2 years. And the range goes, the normal range used to be from southern part of California to the central north part of Chile. And after the 98 El Nino, it reached unprecedented latitudes. It was found in Northern California, Oregon, Washington, British Columbia, and also Alaska. And the question is that what are they doing? What triggers this range expansion of the squid? What are they doing in this ecosystem? As I told you before, they're highly voracious. They prey, they're opportunistic. They prey on everything they find. And based on stomach content analysis, we know that in the Northern California, they were preying on more than 100 different species, and some of them of commercial importance, like Pacific hake, rockfish, and sardine. And of course, this is of economical importance for us. So before the 97, there were only few scientists working with jumbo squid. I was just working a little bit with this species, but then after 1997, there's the number of scientists just increased exponentially. So we have people from all around the world interested in understanding movement, migrations, and many other aspects, reproduction about this species. And depending on their size, they occupy different trophic levels. So when they are small, they can be a very important prey of a huge amount of predators. And as they grow, the number of predators are reduced, but themselves, they are very important predators. So the ones that we found here in the northern part of California are actually the large form. You can find the squid from 50 to 70 centimeters mantle length. And this is a very simplified food web, just to show you the different trophic levels that Dosidicus occupy in the systems. And of course, humans, the fisheries are important. The jumbo squid is an important fishery resource in Peru and also in Mexico. We export them to Asia, where they are consumed by Japan, Korea, as well as China. So we want to understand food webs, reconstruct food webs. And as you can see, they're highly complex. And the main method to understand these food webs is the stomach content analysis that give us very important information about the prey items consumed by each predator. But you can imagine how difficult it is to reconstruct these food webs through time or through space. It's just a huge amount of effort. And at the same time, how these food webs respond to climate, to the ENSO, to the PDO. A lot of scientists have been working on this for several years, and they use models like EcoPath to understand this variability. And another technique is the stable isotope analysis that had been used in paleontology, archaeology for more than a century, and also in marine geochemistry and marine ecology. The oceanographers use them in order to understand the distribution of nitrogen in the oceans. So nitrogen 15 is considered a very important tracer. So by measuring nitrogen 15 values from nitrates, primary producers, zooplankton, particular organic materia, we can understand the nitrogen cycling and identify areas dominated by some processes like nitrogen fixation or by the nitrification where the isotope values are enriched, they are high, or nitrogen assimilation here offshore Chile. And in marine ecology, it has been applied by the 1980s, so about 35 years approximately. And it was found that the differences between a consumer and a prey is about 2.5 to 3.5 parts per mil. So ecologists have used it to understand trophic relationships and reconstruct food webs. But for some reason, these two disciplines just move apart. They were disconnected Oceanographers were not interested in anything bigger than a zooplankton, and marine ecologists overlook this huge variability that we have in baseline values. And as you can see, the values of the primary producers, they change through space and of course also through time. So if you don't have values at the base of the food web, it's difficult to have a proper interpretation of the values that we can obtain. So now you have a value of nitrogen from a consumer, for example. Now you know that nitrogen 15 gives you information of both the habitat and also the relative trophic position of the animal. So probably you have here, you are what you eat plus 3 parts per meal. Okay, that's not true. You are where you eat, because animals integrate that information of their habitat through diet, and also the trophic position. So now, and as you can see, the difficulty is trying to differentiate the effect of trophic from habitat baseline values. But now we have this technique that that instead of one tracer, we can measure isotopes from 12 to 14 different amino acids. And we are still learning about the meaning of each isotopes from each amino acid. But in general, we can classify them in 2 different groups, source amino acids and trophic amino acids. The source amino acids, in general, preserve the values of the primary producers. There's a very small little enrichment with trophic level, and the best source amino acid is considered phenylalanine. While for the trophic amino acid, there's a fractionation. The isotopic values of the trophic amino acid increase due to an isotopic fractionation, and they are enriched, and the best trophic amino acid is considered glutamic acid. So using the values of the source amino acid, we can identify nitrogen sources, and when we have information from the source and the trophic values, we can estimate trophic positions of animals. So one of the first studies that used this technique was in 2002 approximately, and later in 2007 to 2009, some scientists proposed some equations to estimate trophic level of species, and as you can see, without the need of samples at the base of the food web. However, now we know that the trophic level of top predators are underestimated. And currently, there are many scientists around the world trying to understand better the dynamics. So there's some limitations about this technique, but also there's some advantages. And one of them, as you can see, we can differentiate baseline values from diet of the animal. So now, if we have a top predator that is resident from an ecosystem, is sampling our ecosystem for us within its home range. And if we have samples at through different seasons or years, can we track variability at the baseline of primary producers and also at the highest trophic level? So here I propose that sperm whales can be a good ecosystem indicator for offshore ecosystem, and I'm using short-beak common dolphin for more coastal ecosystems. And then we can also understand what is the role of these new species in native communities. How the food webs change. Do we lose some species, some links between predator and prey? Do the food webs get shorter or longer? And those are the questions that I am investigating, and I'll show you some results. So these are the questions that I will address during this talk. Can we trace movement and geographic origin of Dosidicus during its range expansion? Can we infer changes in ocean biochemistry using a top predator as an ecosystem indicator? Can we trace variability in food web length in relation to climate and local oceanographic variables? So first, I'll talk about jumbo squid. And I'll show you results from the ETP because I want you to compare the patterns that we found in these areas in comparison with the California current. This is the last cruise that so far has been conducted in the ETP by NOAA. They are mainly trying to understand the distribution of cetaceans, and I was lucky to be part of that cruise. I collected squid in many stations, so I just selected these ones in order to understand the variation in isotope values between squid between these stations. And we developed a new method to understand ontogenetic changes in diet. So I worked very close with a student. Her name is Erika Villa, and we decided to use for first time gladius. So once we have a squid, we measure the mantle length. This is the ventral part of the squid, and the mantle length goes from here to the tip. You can see here the funnel, and in the dorsal part of the squid, we can get the gladius. It's made of chitin, and as the squid grows, there's addition of chitin. Some scientists use them for age determination, but for us, we decided to cut or measure every 3 centimeter section, pretty much based on funding, and measure stable isotopes for each section. And this is the way it works. So here we have a graph with length along gladius that corresponds to the size of the squid and nitrogen 15 values. And this is my student. It's actually the first student that I have, and she finished her bachelor degree, her master, and currently is working on her PhD. And here we have one line that corresponds to one individual. And as you can see, we can assign one isotopic value per section and understand ontogenetic changes in diet or habitat of the squid. And here are the results of these 6 stations. As you can see, there's a lot of variation and variation between them, but there's a specific pattern. Most of them increase, the isotopic values increase as the squid increase in size. And disagree with information that we have based on stomach content analysis, that as the squid grows from larvae to 30 to 40 to 60 centimeters mantle length, they're preying on prey of higher trophic position. So we were happy and said, okay, now we have this new method. Now we can use this method to understand ontogenetic changes in diet or habitat of any other squid species, including the Archaeopteryx. And actually, I have some samples of Archaeopteryx, but I will not be able to show you these results. So we decided to go to the California current. There was another cruise, which is the Orca well, that again is exclusively for cetaceans, but I was allowed to be part of the cruise and collect a squid. It goes from Washington to California, and it comes back to Washington eventually. So to my surprise, we collected a squid almost in every station here. You can see the triangles and the circles are the areas where we successfully collected the squid, and they are everywhere. They were in the north, south, east, and west. And I pre-selected some stations to extract the gladius and do stable isotope analysis. And I would show you the results. So remember the pattern that I showed you before for the ATP, and now look at the results from the California current. We have the same length along gladius, the same graph, nitrogen 15, and again you can see a lot of variability. But some of them, actually, the isotopic values decrease as they increase in size. Some of them remain relatively stable, and another ones increase as we found them before in the ATP. So one possible interpretation is, well, they're shifting diets, but this is unlikely that a small squid will eat something of lower trophic level as they grow. That doesn't correspond to what we know with a stomach content analysis. So another hypothesis is that potentially these represent 3 different migratory routes. And in order to know if this is true, we selected some sections to do compound-specific analysis. And I'm going to show you the results of phenylalanine that, recall, this is the most stable source amino acid, which means that the values of the primary producers are very close to the Phe values of a consumer. So if we have that these Phe values are different, then it will give us a good evidence that they are coming from different areas. And these are the results. So it's the same graph, length along gladius, nitrogen 15, and we found that indeed the Phe values are different, and when they are small, and as they grow, they homogenize the isotopic values because they are feeding here in the California current system. So number one, we found that they have multiple origins, but where are they coming from? Of course, one good explanation can be from Mexico, from Baja area, where we know that the ETP have high values of nitrogen, of nitrates, and they influence these waters, and that potentially can explain these high isotopic values. But what about these ones? Their values are low, very close to 2 parts per mil. It's actually could be related with areas of nitrogen fixations, and to our knowledge, there's no nitrogen fixation in these waters. So one potential hypothesis is that when the squid move into the California current, as they were feeding and growing, they need warm waters to lay their eggs and reproduce. So where can they find warm waters? Mexico, but potentially also offshore waters. So there's an hypothesis that they move offshore, perhaps lay their eggs, and then as they grew, they came into the Northern California. They stay in this system for approximately 12 years. And initially there was an hypothesis that there was only one single stable population here that were reproducing. And now we know that it's not true. They have to be, there were multiple immigrations into the California current. They were doing the same for several years. And it's possible that they will come back again if we have a strong Nino again. But anyway, that was an exciting result. Other people are tagging squid, but when you tag a large squid, for example, you can only understand one part or one stage of their life, and we're missing the early stages. So with isotopes, we can track this variability from small size to large sizes. And you can use it with other, as I said, other squid species, and even with other predators, like if you have vertebra, if you have teeth of an animal, you can also track these ontogenetic changes in diet and habitat. So now I'm going to talk about sperm whales, and they are the top predator of the Mesopelagic Ocean. They are the most sexually dimorphic cetacean. Males can reach 16 to 18 meters, and females are half their size, 9 to 11 meters. They go to the deep water to feed, to catch their prey. Females usually about 800 meters, while males go even deeper. I think we have an idea it's 1,000 meters, but there's some information that they can go even deeper. We think that they can go even 2,000 meters. And they are found almost everywhere. And as I mentioned before, females remain in the same area most of their life. They can live for 60, actually 70 years, perhaps even more. And while the males, they're highly migratory, and we know very little about their migration. So potentially, females can be good ecosystem indicators. So we decided to go and recover skin samples and then measure stable isotopes to understand changes in the offshore California current. So here is the study. We have samples from stranded individuals and also from free-ranging animals. We have one sample in 1972 and samples from 93 to 2005. And just for comparison, we have here the data from the Southern California Bight with shore-beak common dolphins. And here we have, I'll be talking about this later, about 200 samples. So we measure isotope bulk from bulk tissue and also for compound specific. And these are the results for sperm whales and dolphins. They follow the same pattern that we have found for other species. And you can see here for the glutamic acid, okay, actually first phenylalanine. This is for both the sperm whales and dolphins. They agree. They actually have different habitat, but the variation is very small. And here for glutamic acid, the isotopic values are lower than sperm whales as we expected. And here are the results from the bulk tissue, having from 1972 one sample and from 1989 to 2007. And then if you include this sample or not, we still have a decline in the nitrogen 15 values. So there's two potential interpretations. Well, the sperm whale is shifting their diet. They are feeding on something at lower trophic level. That's one explanation. But another one is that their baseline values are perhaps shifting. And I'll show you the results for component specific. And we have results for carbon, but I'm just going to show for nitrogen. And here we have the values of the average source amino acids. So we have 4 amino acids in each value. And as you can see, the values decline through time. Here we have our sample in 1972. This is the data from the bulk tissue, bulk nitrogen 15, and the trophic amino acids. There's some variability, but for the most part, they follow the variability in source amino acids. So this evidence shows that the values of the primary producers are changing. Why? We do not know. And there's some hypotheses that perhaps the North Pacific gyre is expanding. Therefore, there's more nitrogen fixation, and perhaps the values are decreasing. But so far, based on the data that we have from the sea surface temperature and for the PDO for offshore waters, within our period of time, we have evidence of cooling. So the ecosystem has been cooling and not warming. And that together with this study can suggest that there's potentially more nitrates in the water and the isotope values are decreasing. So this is a study by Rikandjemski and Dunn in which they model the impact of warming and how the nutrient supply to the California current is going to change in response to warming. And they suggest there's going to be an increase in nitrate concentrations and even primary producers with warming. And here's the window of time that we have, and as you can see here, they're increasing. So again, these are just hypotheses. We need data from offshore ecosystems. And even as I told you before, that we have this time series information from different programs, we cannot use the data from coastal ecosystem to explain what is happening offshore. So I think that there are many people working and trying to collect data from offshore waters. And hopefully we can give you more information about this. So of course, I show what we can do with skiing, but we only have 12 years or 13 years. I want more. I want to reconstruct 100 years of data. How can we do this? We can use sperm whale teeth. So these are scientists that collected specimens during the whaling times. They were, sperm whales were the main target of the whaling industry from the open whaling and also the modern whaling. This is Dale Rice that was working here in the California current in the San Francisco whaling station, and Robert Clark in the southeastern Pacific in Peru and Chile. There's 4 different whaling stations together with his wife, Obla Palisa and Anelio Aguayo. And they were glad and happy to say, here, we use it for age determination. We did our job. Here is your turn. So now we can go and collect dentin for each growth layer band. So that's what we're doing. We're using a micromill and measuring the width of the bands. Each band, we think that they represent 1 calendar year. So if we know the year of death of the animal, this is the bulk cavity, we can reconstruct the age of the animal and at the same time the isotopic values and understand variability through time. So here you can see how we are collecting the sample. This is for a male. And I'm going to show you the results for the California current for females. Before that, as any project, there's always some issues. So our micromill broke, our funding ended. So I will show you just the data from the bulk record. And now I'm actually trying to get funding to do component-specific data and understand the patterns. So here are the results. We have time from approximately 1938 to 2008. Here we have data from 6 or 7 females. We have more, of course, from before 1970s during the whaling time. and only one female that was stranded in 2008. So if you guys find a stranded female, please let me know, because I'm very interested in getting, analyzing more samples. So as you can see here, there's a decline, a slight decline from the 1940s to the 1970s, and the decline is actually increased after the 1960s. So again, what is shifting? Already we have the information that they can be baseline values, but look at this. It's a sharp decline coincident with the 1998, 1990. This is El Nino, a strong Nino. And then 1997, 1998, 1999. So there's some variability there. But also it can represent that the well was moving. So, however, let me show you the data for carbon. Carbon, it also gives us information of primary productivity, and it's the same animals, the same record, and as you can see, there's an increase, and then a decrease after the 1970. But look, the variability is only within 1 parts per mil. That gives me some clues that the wells remain in the California current system, and so I am more interested to see what is happening here. What is this? So to be continued, hopefully I can show you results in a year, less than a year, and tell you what's happening. But this gives us evidence that what we have found with the skin, we can have a lot of information also with heat. We have results from the Southeastern Pacific Ocean, from the from the Humboldt Current and the EPP, and we do not have data post-1970s. And before 1970s, we do not see the same patterns that we have found here in the California current. So now I'm going to talk about food webs, variability in food web length, and I'll be talking about dolphins in the Southern California Bight. So our dolphins, they are found every year in the Southern California Bight. Of course, they move north, south. We collected samples from 1991 to 2008. In total, we have 203 individuals, and they are incidentally killed from the gillnet fisheries. So we know the cause of death, and we know that they are healthy. We have information from length, sex, and age, and of course, the NOAA scientists recover all possible samples and information, and they make it available for me for isotope analysis. So I analyzed nitrogen-15 for 203 and component-specific for a few of them. Just a small window information. The cost of component-specific is more expensive. It's about $200 per sample, and some labs charge about $500 to $800. So I would like to analyze more samples, but funding is limited. So we use nitrogen to help us to understand the pattern, and then select some samples randomly to conduct component-specific analysis. So here are the results, time and nitrogen-15 values. There's a lot of variation there. And you can identify a pattern. There's a potential decrease from the 1990 to the 97, 1998, and after that, an increase in the isotopic values. But again, we really don't know what is changing, diet of the dolphin or baseline values. So now I'm going to show you the results from component-specific analysis. I will show you 4 different records, and then actually 5 different records. So here's the results of amino acid. We have time, we have nitrogen-15 values, and here we have the results for phenylalanine, Phe, that remember is the main source amino acid, and then glutamic acid. You can see a lot of variability through time, and there's some oscillation there, but also the trophic amino acid, they are changing. And as you can see here, a sharp decline coincident with the 97, 98, 99, and then a recovery of the isotopic values. We decided to also look at the data for average source amino acid and average trophic amino acid and see if we find the same patterns or not. And here are the results. We have time and we have nitrogen-15 values, and indeed source amino acids, they show the similar pattern that phi, and the same with average trophic amino acids. And here again, we have the low decline in the isotopic values. So something is going on there. But then I would like to understand the effect of these different indexes, right? So in the Southern California Valley, as you know, we have the Cal Coffee program, so we can have that information in order to model and understand this variability. And also we have climatic indices like the PDO and the ENSO. But before that, I decided to measure, instead of just modeling phi or GLU or source and trophic amino acid, I want to measure the distance between the trophic and the source amino acid, between GLU and phi and trophic and source. And that can represent a proxy of food web length. So we have already the top predator, the position of the top predator and the source amino acid. And then we have glutamic acid and phenylalanine. Okay, so these are proxies. And then we can model how they change in response to climate, right, or environment. And I'm going to show you, I have a lot of oceanographic variables. So I actually started with about 16 variables. They have a lot of data, and it was overwhelming. So I was like, woo, okay, let's choose those that I think, or we think that were more important for the dolphin, for the food web, or for the prey of the dolphin. And also, of course, the climatic indices. So we have the MEI, that index for El Niño Southern Oscillation, the PDO, and I will not show you results for the MPGO. We select only these two. I'm going to show you data from the availability of nitrates. We have records of sea surface temperatures, but I thought it would be a lot, so I just would show you data from 4 different predictors. And these are the results. Again, we have bulk in black, the differences between GLU and phi, the difference between trophic and source amino acids. And there's, number one, you can see that the proxies of food web length, they do not agree with the bulk record, right? But there's an agreement between GLU and phi and trophic amino acids for most of the predictors, with, of course, some, except for the PDO. And here we can see that for the MEI, for example, we can see that the food web length, or the proxy of food web length, is short during positive values of MEI and also negative values of the MEI. This one corresponds to El Niño and La Niña event. And the food web length is longer during average conditions. The same for AN. AN is an index of the available nitrates in the euphotic zone. For the PDO, we have a different pattern where the food web length is longer during a warm phase as well as the cold phase of the PDO, and shorter during the average conditions. And for nitrate concentrations, you can see that the values, the food web length is shorter when the concentrations of nitrate is higher, and longer when the concentrations are low. So ecologists have tried to understand the variability in food web length for almost a century, but we know a lot about terrestrial ecosystems and also lakes and some experiments, but we know very little about marine ecosystems. But there's a paper by Seki that suggests that the food web lengths are shorter in highly productive ecosystems and longer in oligotrophic waters. Why? Because the upwelling ecosystems are dominated by phytoplanktons, by diatoms that are large. Zooplankton feed on these diatoms, and therefore the number of steps in the food webs are shorter. If that is true, disagree with this pattern, right? When we have shorter food web lengths, when we have high production of nitrates, and longer when the concentrations of nitrate is lower. But what about this or this one? Why do we have a different pattern between the PDO and the MEI? And there's many hypotheses, but one of them is the hypothesis of disturbance that suggests that when there's a high disturbance, there's a removal of some species, and therefore, especially if we remove the strong species, the number of food webs are reduced. So the higher the level of disturbance, the shorter the food webs. And these are in agreement with this pattern for the MEI. And if we think about this, the El Niño, La Niña, they last for 1-2 years, while, for example, for the PDO, that we have a different pattern, we have decadal scales, a variability every 10 years. So I'm suggesting that perhaps during these PDO phases, that the communities are more adapt to these conditions. There's enough room for them for the food webs to increase, and perhaps that may explain this pattern. I'm very excited about these results. Again, and of course, you guys some ideas. I welcome your ideas, but this gives us a very good a picture of how the food webs, they are not stable through time. They change, and then we can have a good understanding about how they are changing. And again, this is just for the Southern California Bight. It will be interesting to see if we can find similar pattern for offshore waters or for other coastal ecosystems. So in summary, I show you that component-specific analysis, number one, can help you to decouple trophic effect from baseline values. It can be a very important tool to determine movement or migrations of and geographic origins of highly migratory species. We can track changes in biochemical recycling and animal trophic status using top predators as our ecosystem indicators, and also can provide proxies of food web length to evaluate their variability through time in relation with environment. And I think that's everything I was going to show you. And of course, we have had funding from NOAA and NSF. Thank you. I'm happy to take any questions.
For the
food web length?
Yeah.
Yeah, I mean, we have data. I mean, when you have a predator, what we want to know is that what is important for them, right? So we are trying to understand what is happening in the euphotic zone. So most of the data is for these first 200 meters. And for dolphins, they can dive about 200 meters as well. I think that's the range. So, and then I know that there's many species that migrate. They make these diurnal migrations, but we are just currently just getting the data from the euphotic zone and model that in relation to this variability. I don't know what other species you think. We think that we're covering the main prey items. So the main prey items of dolphins are squid, myctopids, what else? Octopus and so on. Actually, there's a student working with stomach content from the same individuals that NOAA has been obtaining, and they're like about more than 60 different prey items. I don't know, or I cannot think about any what prey can be so important that I can't include it in the model. But we have data from the small phytoplankton volume. I did not show the results. We removed the effect of jellyfish because we thought that it was not important within the food web of dolphins, but that's the selection we made.
So the sperm whale basically are deep.
Yes.
They eat deeper in the water, like kind of going back to Chip's question.
Oh yeah, okay, yes. So they, I'm sorry I didn't talk about that. Sperm whales, the main prey items are deep water squid, including the architeuthis. they can also prey on cod or on fish at higher latitudes. We believe that cephalopods, like Tosidicus, for example, or other Omastrepis and Nicoteutis, they rely on the information from the euphotic zone, and they are not dependent for example, hydrothermal vents. So that's why even if they are coming from the deep ocean, they still are dependent from the euphotic zone. So while we're tracking, even if this is a mesopelagic top predator, we still think that we are recording what is happening in this layer close to the surface.
I'll keep
Yes.
asking then. So this, I mean, the two major squid that you've been talking, well, one squid you were talking about, Humboldt squid, obviously really fast growing, 1 year, 2 years maybe.
Yes.
And so a predator eating one of those is eating the signature that's associated with a 1 year cycle too. But do you know very much about like some of the deep water squids, how long they live and whether they can be changing their isotopic values over integrating them over a longer period of time as
Yes.
opposed to say Loligo or Homostrepes?
Yes. For squid, I mean, if we use, for example, mussel, the turnover rate is very fast. I used to think that was 2 months, but actually it's faster than that. We actually think it's weeks because they are just growing. They're just eating and growing and reaching more than a meter of size. For deep water species like Echinocoris, I think there's still controversy about how old they can be. And I think there's controversy. Some of them think that 3 years, another one 6 years. But imagine the size of this squid. I mean, the age, 6 years for this large size. They are about 12, 14 meters. When you have a mussel, I cannot answer your question. I need to know how fast they grow, and in order to have a better estimate. But if you have gladius, as the squid grow, they add chitin. So if the squid lives for 6 years, I have 6 years of information in the gladius. So you guys find Echinocoris, just let me know. Actually, there was one individual stranded here in Santa Cruz in 2008, and I got some samples. Unfortunately, because it's a specimen, it was sent to a museum, Santa Barbara, I think, and they're really picky. They don't allow you to get all the samples we want. We were able to get only a few of them, but not enough to reconstruct the whole variation in isotopic values. They actually show a belt like that. I mean, if I recall the nitrogen values, they are like this. We think that they are not part of this system, this cold waters of the California current. They are found mostly in subtropical waters. So what we think that happened, it maybe migrate, move into this California current, and eventually, they move back. We really don't know too much about the giant squid, but that's one of the hypotheses. There are some records in many places around the world, especially if you follow sperm whales, you can find giant squid in Japan, and I think the Japanese have many specimens for Echinocoris. Also in Australia, in New Zealand, there's good specimens there, but for the museums, of course, they need a lot of space in order to keep these animals in the climate. But yeah, there's a lot to do. You can analyze the same with squid beak. It's more difficult because how to assign the isotopic value for a specific size. With gladius, we know that the length of the gladius corresponds to the natural length of the squid, but the squid beak is more challenging. It has been done, and if that's everything we can have, we can understand the variability. Yes.
Is there much variability from year to year in the ambient nitrate isotopes in the Southern California bite, or is it relatively constant?
Yes. I actually no. Sorry, I didn't include. Oh yeah, I did. This is I have it ready. In case you guys ask that question. So this is a study from Greg Law, collaborators. that analyzed nitrogen-15 from zooplankton, 4 different species of zooplankton in the Southern California. And they tried to understand how this variation in nitrogen values are related with the variability in nitrogen in this area. Again, it's only bulk nitrogen-15. They have almost 50 years of record. Unfortunately, they don't have too much data here in between 1980, 1990, and the 2000, when we have the strong Nino events. But you can see, again, there's a lot of variation. They modeled these values from these 4 different species in relation to climate as well, PDO, ENSO. They found some relationships for some of them, but it was, they use, I think, mainly linear regressions or linear models. So we don't, they didn't see too much a trend that was in agreement for all of them. It's only one species that I don't remember which one declined through time. The isotope values declined through time, while the other ones there's a lot of variability between them. And the main explanation was because there's a lot of variation in the concentrations of nitrate and even others like silica and other nutrients in the water. If we have information from other ecosystems that there's huge seasonal variability in the nitrogen value. So you have, if you measure nitrogen from primary producers or zooplankton through time, within a year, the variation changed in the summer versus the winter. Animals, when we're working with animals, depending on how fast they grow or the metabolism, we have information from weeks to 2 months to 3 months. If we have soft tissues, even at 6 months, but if you have like shells or like hard tissues, we can have information for a longer period of time. And, but yeah, did that answer your question? Yes. More questions?
Well, she'll be around here. It's not. Does anyone have any questions? All right, well, thank you very much. And she'll be around, so if you come up with more questions, she'll be able to answer that.
Thank you.